Hey guys, welcome to Chapter 12, Topic 1. For as easy as Chapter 11 probably was for you, Chapter 12 is going to be a lot more complicated because we're taking the concepts that we just talked about in Chapter 11 and applying them in Chapter 12. So I expect this chapter to be a lot more challenging for you, so please let me know if you have any questions. Chapter 12 we're going to divide in two parts. We're going to talk about transporters and the basic properties of membrane transport in Topic 1. And then in Topic 2 we're going to talk about ion channels and some examples of how nerve cells work. So those are how we're going to break this up. As I said, in this topic, we're going to talk about the properties of membrane transport and then examples of transporters. Here are our objectives. As always, if you have any questions about any of these, please let me know. I expect you to have all these accomplished and understood by the time we move into our next exam. So let's talk about the properties of membrane transport. In the last chapter, we talked a lot about how membranes are responsible for separating the inside and the outside of the cell, or inside and outside the organelle. What we didn't talk about was how we actually move things in and out that we want in and out. And so it's important that we understand how this works. So we're going to talk about concentration gradients, the selective permeable barrier, and then types of transport and transport proteins. As I talked about with osmosis in the last chapter and all these concentration gradients matter a lot. And so it's really important if you're feeling murky on concentration gradients, that now is the time that you get some help on that. And let me know if you need some help with me, or you can see the tutoring center. But concentration gradients is what drives a lot of the functionality in the cell and movement across the membranes. And so it's really important to understand how that works. And so what I have here is a chart that shows you some of the ions that are frequently inside or outside of the cell and the concentrations that you can see on them, and you can see how we have the gradients. For instance, sodium, there's a really high extracellular concentration but a very low intracellular concentration, and potassium is the opposite. So I want you to take a moment and look at this. I don't expect you to memorize this chart, but I do expect you to understand which ions are in greater concentration outside the cell than on the inside of the cell. So spend some time looking at that and understanding that component of it, because this is what's going to drive these transports we're going to talk about here in just a second. So that membrane, we keep talking about it, it's a selectively permeable barrier. So it lets some things pass through easily and it stops other things from passing through. Small hydrophobic molecules pass through easily: water, CO2, N2, benzene, etc. And small uncharged polar molecules also do. So glycerol, ethanol, water, etc. But anything that gets bigger has to be transported across the membrane. The same goes for anything that's charged. And this, as you should know from your chemistry classes, is because of the way the chemistry of the membrane works. So when it's charged, it's not going to be able to pass through, or if it's too big, it's not going to be able to easily diffuse through. So it's important to understand how that works, and I expect you to understand these four groups of molecules and be able to identify which ones would be able to move freely across the membrane and which ones wouldn't. So that leads us to the types of transport. How do we get those charged ions and those big molecules across the membrane? Well, we have two main types of transport. We have passive transport, and these are molecules that will just move down the concentration gradient. There's a lot on the one side. It will easily pass through these transporters to the other side through an ion channel or a transporter. Active transport requires energy or something else to help it move against the gradient. This is when we want to pump something out of the cell that is low in concentration inside the cell and there's a higher concentration outside the cell, or vice versa. We're going against that concentration gradient, and it almost always requires energy or something else in a coupled reaction to help it move. So we have two main types of transport proteins, and we're going to talk about transporters in this topic, and we're going to talk about ion channels in the next topic. So you can see here that transporters are proteins that have this conformational shift that's going to move a molecule across the membrane, whereas an ion channel is more like a pore and allows things to move through the cell like that. So it's important that you understand how that works. Transporters have a little bit more control over directionality of the transport, whereas ion channels are a little bit more have a little bit less control of it, because as you can see, the directionality is not completely controlled in this environment. And there's some components that do control how things flow, but for the most part, they don't control if things are moving in or out. They're just control whether or not they're open. And as I said, we'll talk about those in the next topic. Whereas transporters are directionally specific, usually. So it's really important to understand how that works. So let's talk some more about transporters. And so in these next couple slides, we're going to talk about what drives them, and then we're going to talk about a whole lot of examples. All of these examples, I expect you to be able to know and understand and discuss in depth. So make sure you spend some time with all of these examples we're going to talk about in these next couple slides. So as I said, there's two types of transport that can drive transporters. We have passive transport and we have active transport. These first two examples are passive transport. The first example is concentration gradient. This is just like that sucrose bag that you filled up in B201, except for this time we're going to use proteins to help move things across the membrane. So what you can see here is the solute will bind to the transporter, conformational shift, and it will release on the other side. This is driven by the high concentration gradient outside the cell. We also have electrochemical gradients. There's a charge that runs along the outside of the cell, and all the cells have these, some to a greater extent than others, but these charges have this. And so the charges can work in concert with the concentration gradients, as you can see in example A, where when there's a whole lot, when the positive charge is on one side and the solute is positively charged, it will allow for the pumping of the solute across the membrane through a transporter. If the electrochemical gradient's the opposite, then it does work to the same extent, but to a lesser extent. And so you can see how this charge will work about that. And I know this electrochemical charge might be a little murky, but hopefully we'll clear that up here in just a second with that. So as I said, active transporters, these guys require energy or something else to help them go. First one is a coupled transporter. This is where we use the concentration gradient of another solute to pump a different solute in or out of the cell. This is essentially a coupled reaction we talked about, except for energy is not being moved around. It's just piggybacking on the system. Then we have ATP synthases. These are proteins that use ATP, break it down to pump things across the membrane. And then we have light-driven pumps. And these are like the rhodopsin we talked about in chapter 11. These are pumps that use light energy to help drive things across the membrane. So let's look at a couple examples now. Okay, so here's our sodium-potassium pump, and you've probably heard about it before, but never discussed it totally in depth. If you remember, or if you've printed out slides from before, you can see that sodium has a huge concentration gradient. There's a lot more sodium on the outside of the cell than on the inside of the cell. Coupled with that positive charge that runs across the outside of the cell is forever in this constant battle to keep sodium out of the cell. It doesn't want it in the cell, and the outside environment just wants to force more and more in. And then, conversely, the cell wants to keep a lot of potassium in the cell. And there's not a whole lot of potassium outside the cell, so it's going to grab anything it can to bring it in. And so what the cell is going to use is the sodium-potassium pump. And this is an antiport, so it's going to pump one thing out, one thing in. And as you can see here, the sodium is going to enter in, phosphate is going to be added through the breaking of ATP down. This high energy phosphate is going to change the conformation of this enzyme. It's going to release that sodium to the outside of the cell, which then it changes that conformation, allows potassium to bind, brings potassium into the cell, releases the high energy phosphate, and restarts this entire process. So it's using that ATP to help pump sodium out against the electrochemical and the concentration gradient and bring potassium into the cell. So it's really important to understand how this works. This pump is a major component and a major energy user of the cell. It uses about 30% of all the ATP the cell makes, trying to maintain this osmotic balance. So let's take a look at that. So this osmotic balance, as we talked about, this is critical because what happens is we have to keep the sodium levels low inside the cell so that the cell doesn't swell and burst. So this is why the sodium is constantly being pumped out of the cell, because we got to keep these concentrations fairly even, or we have to keep these concentrations set like this so that the cell can keep the water from rushing into the cell and bursting the cell. So it's really important to understand why the importance of that sodium-potassium pump. Another example, and this is a unipump, or a uniport, is the calcium 2 pump. Now it works in pretty much the same way. As you can see, ATP comes in, uses the breaking of that phosphate to cause a conformational change, which releases calcium into, in this case, the sarcoplasmic reticulum, which is similar to the endoplasmic reticulum, but in cardiomyocytes. It's a very similar process, conformational shifts and everything like that, but it's a uniport, so it's important to understand that. Calcium is a huge player in our cells. We have to have a lot of calcium, and we have to be able to move it around in order to have a variety of things function, including our hearts. So it's really important that you understand how calcium is moved around. So that's why this example is here, even though it's very similar to the sodium-potassium pump. All right, so let's talk about the glucose-sodium symport now. So in this case, this is a symport. We talked about a antiport where two things are pumped in opposite directions. We talked about a uniport where one thing is moved. Now we're talking about a symport where two things are moving in the same direction. Now in this case, the cells are utilizing glucose, or the high concentration of sodium, to move glucose into the cell. There's a low concentration of glucose outside the cell, so the transporter uses that concentration gradient of sodium to drive the conformational shift, which allows for us to pump more glucose into the cell against its concentration gradient. Now let's look at how this works in the body. So we have the cells that line the lumen of the gut. Now inside the gut lumen we have low concentrations of glucose. We couple this with the sodium to bring it into the gut cells, which obviously should have a higher concentration of glucose because they're right there where the food is being broken down. So they have a higher concentration. Now that we've used that coupled symport, we can then allow for passive transport of that glucose out of the high glucose concentration of these gut cells into the extracellular fluid and shared with other cells. So this is how this possibly works, is that we use active transports to allow for passive transports to spread nutrients and things like that throughout the entire body. It's a pretty cool process. And so it's really important that we understand how this works. And then obviously you can see here at the bottom that sodium-potassium pump shows up again to pump out that extra sodium. Another thing that's important to understand before we move on is hydrogen gradients. We're going to talk about these proton gradients when we do in chapter 14, when we're talking about photosynthesis and we're talking about cellular respiration, the electron transport chain. But it's important to know that cells use these too. And they use them for a variety of activities to help induce passive transport. They use active transport to make passive transport possible. And you can see some examples here. Prokaryotes and fungi and plants use this a lot. Animal cells, not so much, but it's important to understand how this works. And so it's a very similar process. They can use ATP, pump out the protons. And then that high gradient of the protons allows for those solutes to be pumped in. Now, it's also important to realize that cells will pump protons into vacuoles or other organelles where we need to keep the pH low, so that the pH is low so that it'll function properly for that organelle. So here's just a quick review of our transporters. We have some examples of the uniport, which remember is one thing going in one direction, antiports, which are two things going in opposite directions, and symports, which are two things going in the same direction. I expect you to be able to discuss all of these examples that we talked about. So if you have any questions about any of these, please let me know, and then we'll go over them some more in class.